I hope you can see my screen now. Um, Fantastic. Um, I will introduce a technology to you called expansion microscopy, and in particular, how we applied expansion microscopy to plants for the very first time. And I would like to point out this is very much the work of Michelle Gallet that I'm presenting here. She was an extremely talented PhD student at the Institute, did a lab rotation in our group, and well, as Alex already disclosed, I have no idea about plants. I've never worked with plants. I only know how to water them in my house, but that's about it. And Michelle is a real expert in Arabidopsis biology, and so we tried to see what can we do together. Can we combine expansion microscopy to use it on plants? And this is how this whole project came about. A little bit about what we actually do in my group. Most of the time, we do brain mapping. I'm recently established this group at the Laboratory of Molecular Biology. And basically, we use expansion microscopy to read out the circuit architecture and the morphology of neurons. And then we apply various molecular tools to get a molecular readout of the properties of these neuronal networks. And then ultimately, we put it all together and arrive at a better mechanistic understanding. But I promise this is it for neurobiology for today. The rest is all plants and expansion. So, first of all, what is expansion microscopy? So you can see, I guess I lied, you can see a brain slice here. And the brain slice is growing as we are exchanging the water in this little dish here. And this is literally what expansion microscopy does. You take a biological sample and you increase the sample in size up to nowadays 1,000-fold, most of the time much less. It just depends on how much resolution you require. So why is this useful? Why do we want to increase the size of a sample in the first place? So imagine you have a fluorophore, a point emitter. So it sits here at the center, the little dot, and then it emits fluorescence all around it in a sort of spherical area. This is fine as long as you have only one fluorophore. But if you have two fluorophores and they're very close together, these emission circles basically will overlap and you cannot tell them apart anymore. So this is the big resolution problem that we are dealing with in light microscopy. And this has been described already in the 1800s by Ernst Abbe. Your resolution, the distance that 2 points can be resolved at, is just defined by the wavelengths divided by 2 times the numerical aperture of your objective. So the problem then, of course, becomes if you have two different structures, in this case, bassoon, a presynaptic protein, PSD95, a postsynaptic protein, they're very close together. You can't hardly tell them apart. And this is where expansion microscopy comes in. The simple idea is you just separate these fluorophores until they are far enough apart that you can resolve them. And this indeed is what happens. You expand the sample fourfold, and you can already see the separation. You expand the sample tenfold, and you can see very good separation. And you can now observe these proteins independently from each other. So we just have to adjust this formula from the 1800s. It's the same formula, but we simply divide by the expansion factor, and that's your new resolution and the new level of detail that you can observe. So there were two papers coming out that sort of commented on these publications. And again, people like Laura's technology thought this was kind of magical. Here's the Mary Poppins review of our manuscript, and then there was the Alice in Wonderland review of the manuscript. So people, considered it kind of magical, but I hope that I will show you this is actual science and actual working technology. And I would also like to point out there have been actually two papers back to back who figured out how to use expansion in plants. One paper is from Magali Grison and Manuel Bayer in the University of Bordeaux. And the second paper is from our team at IST Austria. And like I said, the real driving force for this was Michelle. And then we also had actually Alex Johnson involved in this project in the group of Yeshi Friemel and me in the group of Hans Danzel. So, whenever you want to cite this and whenever you apply the technology, I would really encourage you to actually look at both these manuscripts because they're basically really introducing the same kind of approach with slightly different protocol. So, how does it work then? How does we apply this technology? So it starts with a plant root. In our case, we focused on the root of Arabidopsis. We label what proteins we are interested in, and then we anchor those proteins, which means we attach a covalent bond to the proteins that will serve as an anchor for the gel polymer. And then indeed, the next step is we form a hydrogel polymer that covalently interacts with those anchors and retains the labels that we put in. Then we digest away the cell wall. This is the most important thing. The cell wall is very mechanically resistant. We need to, as we actually saw in the previous presentation, it's a stiff structure. And if we want to expand the plant homogeneously, we need to get rid of this stiffness. Then we digest away all the other proteins to again soften up the sample. And finally, we can expand the sample. It physically increases in size, and we can look at the details. So, how does this look like in practice? This is the tip of a Arabidopsis root, and you can see here a tubulin labeling. This is before expansion, and you can see, well, there are some structures visible, but not a lot of detail. This is the same root tip after expansion. And this is to show you both the physical increase in size, this is to scale, and also the increase in resolution, because suddenly we're able to actually see the individual microtubules that are making up the cytoskeleton of these cells. So what we achieve with this approach is approximately 60 nanometer resolution. Again, the picture that I showed you before. And now we determined our expansion factor on average is 3.9-fold. There's always a little bit of variability. And then we zoom into this one cell that I'm magnifying here. And if you look at this line scan before expansion and after expansion, suddenly instead of one big strand of microtubules, we can determine these are actually three separate microtubules. So this is great. We are now able to see much more detail. And what about distortions, though? We are expanding the sample. Can anything happen to it? Could there be any effects that we are introducing? Well, we looked at this very carefully using immunostaining for PIN1, a protein at the membrane of these cells. We looked at it before expansion and after expansion. Then we overlaid the two. So in magenta, you see the pre-expansion, in green, the post-expansion image. And then we determined, are there any distortions? And yes, there are some distortions. They do not perfectly overlap, but they are very minimal, those distortions. So over a measurement length of 100 micrometers, you get only about 1 to 2 micrometers of distortions. So this is quite manageable. We have also done this analysis on the tubulin staining that I showed you before. And again, there are some distortions, but they remain rather minimal over the distances that we are able to measure. Also, you can apply these scripts that are all on GitHub to actually correct these distortions if you're very worried about them. So the important thing for this technology, as I mentioned before, is to digest the cell wall. We have optimized this protocol for Arabidopsis, but it is quite possible that for other plant cells, you will need to optimize a different digestion protocol. What we actually hit upon is to just use nature, because nature has already figured out how to get rid of plant cell walls. There are all these different fungi. My favorite is probably the Aspergillus oryzae, because that's what you use to make sake in Japan. And all of these different fungi, they digest plant cell walls to eat the plants essentially. So we mixed together a mixture of all these different enzymes, applied it to the plant that was embedded into the hydrogel, and that gets rid of the cell wall. So this is indeed a common theme in expansion microscopy. Anytime you apply the technology to a different sample, you need to find the correct homogenization conditions, which typically just means you need to find the correct mixture of enzymes and heat, and then you can expand almost anything. So, lesson to take away, you need the homogenization, the digestion with the enzymes to actually expand your plant cells. So, I mentioned before we did 60 nanometer resolution, or we achieved 60 nanometer resolution, but could we do better? And yes, we can. We combined expansion microscopy with stat microscopy, which is an optical approach to super resolution microscopy. And stat microscopy works in the following ways. Imagine all these dots are individual fluorophores. So usually you shine light on these fluorophores and you hit a bunch of them because you cannot focus light infinitely small. It behaves like a wave. So you always get a blurred out spot no matter how well you focus. So you will activate multiple of these fluorophores and all these fluorophores will emit at the same time. And what you get is a big blurry mess. In stat microscopy, again, you illuminate everything at the same time, but then before these fluorophores actually get to emit their light, you apply a donut-shaped depletion beam. Any fluorophore that gets hit by this depletion beam is knocked down to a lower energy state and actually can no longer emit. And the only fluorophores that emit are the ones that are in the middle of this donut. So you get a much more precise picture. So here is plant X. We are here labeling transport vesicles of the Golgi apparatus. And this is the same picture after stat microscopy. So you can see much higher resolution. And this is the same in SET, so the same picture, but now in ZX and not XY. And again, applying stat microscopy leads to a vast improvement in resolution. So you can see this also in this little video here. Very nice precision to localize the individual vesicles. And if you look at this little frame here, it actually is biologically informative. Even with PlantEx, we cannot separate all structures, but with PlantEx and STAT, we can tell these are indeed two vesicles that are touching each other. So we have now used this technology to quantify cellular ultrastructure. You again see here the vesicles, these Golgi transport vesicles. We applied a computational script to localize the center of each of these transport vesicles, which is now shown as the green dots. The green dots are forming clusters by proximity, and we have just colored them in here to identify the different clouds of vesicles forming around different Golgi stacks. So now you can really get to a nice quantitative description of the cellular ultrastructure. We can also use this very nicely for volumetric imaging. Volumetric imaging, especially with super resolution, is often very difficult, but the expansion microscopy leads to a clearing of the sample, so we can image very deep tissue, which we could not do otherwise because the tissue scatters the light. So this is the cluster analysis that I showed from the video, and this is the same cluster analysis encoded by imaging depth. So the different colors represent different imaging depth. And as you can see on the scale below, we imaged about 100 micrometers into the sample. So we can basically go through the thickness of an entire root tip now and get the same level of resolution throughout the entire tissue. And now, even more exciting, multicolor super resolution imaging. With most super resolution technologies, multicolor is very difficult. Stat microscopy is often optimized for just a single color. But now we can do this on confocal or epifluorescent microscopes, any conventional microscope that you can access, and get multicolor super resolution imaging. So in this case, we have tubulin and MAP4 GFP associating with the tubulin. And you can see the two of them associated together. We can also multiplex. This is an example not from PlantEx, but from neuroscience. This is a synapse again. And we have multiplexed here for 6 different proteins. So you could do the same conceivably in PlantEx. We did not do it as part of the original publication, but we are now accessing this multiplexing technology where you can actually do dozens of markers in the same tissue. But let's look at something more exciting than tubulin. We are looking here at gamma COP and VHA-A1, which is a marker for the Golgi apparatus and a marker for the transport vesicles budding off the Golgi apparatus. When we look at this with without expansion microscopy, you see it's very difficult. These are very closely associated markers. They all blur together. So now let's look at them with expansion microscopy. Again, already from a distance, everything looks much crisper. And now let's zoom in. You can now see the individual Golgi stacks here. And then you can really see the individual vesicles, which are forming these clouds around the Golgi stacks. So now we get to a real multicolor representation at high structural accuracy of these transport pathways. We can also visualize very nicely the cellular context. So far, everything I showed was immunostainings, but we can also just do a pan-protein staining. We can label all the proteins that are present in these cells. And this looks somewhat like this at low magnification. So now you see anything that is darker has a higher protein density. We just attached NHS Alexa 488 to the lysines of the proteins. So now if you look at individual cells, you can see now the outline of the nucleus, you can see the outline of the cell walls, you can see these dark areas which are representing mitochondria, the lighter bubbly areas are representing vacuoles, and now we can overlay the immunostaining with it, and we get this very nice colocalization and high structural fidelity analysis. This almost looks a little bit like electron microscopy because, again, we are looking at protein-dense structures as you would do in electron microscopy. But this is all light microscopy, the imaging. So with that, I come to the end of my presentation. I would like to thank the people again who were involved in this. In particular, Michelle, who is now working at Illumina, Alex, who is now at Exeter, and then Yeshi and Hans, who are still leading their groups at IST. Would also like to thank all the collaborators that I have in my own group now, and in particular the LNB and the Global Talent Fund here in the UK, who are basically sponsoring my group and buying all the equipment currently to set up a new lab. Thank you very much.
Oh, thank you, Sven. That was awesome. I have to admit, when people started first talking about expansion, it did just sound magical. Let's just make it bigger. And it works quite well. Before I read the questions, I just want to ask quickly, I mean, obviously you're limited to 4x, and people are starting to do these ultra structural expansion and even things like landscape expansion. Do you think these are translatable over to the plant samples?
Yeah, indeed. The nice thing about expansion is it's a very modular technology. So whenever one part of the field makes any improvement, it's typically quite straightforward to port that improvement to other model systems. So for plants, as you said, we have only done fourfold expansion by now. But I think it would be almost trivial to do at least tenfold expansion, at which point you would not need stat microscopy anymore. You would get the same resolution just from a conventional microscope. And potentially you could go even higher. But yeah, the only thing is that we have not done it because my group focuses more on neurobiology. But anyone who wants to try it out, I would be happy to lend some expertise. I'm sure it would work.
Okay, there's a question. Hi, what type of objective lens working distance should be used when imaging a root undergoing expansion?
Right. So it depends mostly about how big you make your sample. So obviously, expansion microscopy means everything grows bigger. So you need higher working distance objectives if you want to image the entire thing. The pictures that we showed here, they were obtained typically with a 40 to 60x objective with a working distance anywhere from 300 to 700 micrometers. So that is still enough to get the entire root visualized. But obviously, if you had higher expansion, you would need higher working distance. However, there are tricks. You can actually section the gel as you would section fixed and embedded tissue after expansion. You just put it on a vibrotome and you slice your gel. And then you can just image the slices individually and reconstruct it later.
Okay. I think in the interest of time, we will email Sven the rest of the questions. And I hope you can see I'm sharing your screen. So if you enjoyed Laura's talk and you want to learn more, we actually have a BioBrillion conference in Exeter in December. So come and find out a lot more about the technology and talk to physicists trying to understand the maths and making it more usable. Oh, yeah, I don't know why that suddenly appeared, but there's a nice picture. And yeah, and finally, well, thanks everyone who attended, and again, fantastic talks from Sven and Laura. If you want to give a talk, please feel free to sign up. And also for the next session, which isn't July, I've got the date wrong. What date is the 15th of October, I believe.
October, yeah.
Yeah, that should be the 15th of October. But we have Marcel Dickmans talking about in situ cryo-EM, and we're still looking for other people to talk. And yeah, thank you very much for attending. And if you have any questions, please feel free to email us or the speakers directly. Yeah, so thank you very much, everyone.